one, this one is about pH, calculating pH. You can make sure you have a calculator with you. And so the question is, you have to calculate the pH of this thing. So pH of 0.1 mole per dm cube of this acid. Remember, what is K? K is the dissociation constant of this thing. So K is what is K? K is products over reactants. It's H+1 concentration multiplied by the other product concentration, which is Cn-1. And you're going to divide it by the acid concentration, which is HCN in this case. And remember, I told you that both of these ions will have exactly the same concentration. So your expression becomes H+1 squared divided by the acid concentration, which is HCN in this case. So if I solve for Ka, the value of Ka is already given. That's 6.2 times 10 power minus 10. H+1 squared, I'm not sure of. The acid concentration is given over here. It's given as 0.1 mole per dm cube. So that's given. So remember, this is the expression for any K. Whenever you have a weak acid, the weak acid dissociates in the same way. It produces like the general equation was, if you remember this, the general equation was that any weak acid would dissociate to produce two ions. One of them would be H+1, and there would be some other random ion that's going to be produced with it. So these two ions are going to get produced, and you always get this expression, H+1 squared divided by the acid concentration. Can you even quickly tell me what is the concentration of H+1 that you're getting? As I started off with the paper, I think we were supposed to do, sorry. I think we were supposed to do nitrogen compounds, which we were doing actually. And we were going to complete organic. Just a second. Does anyone remember does anyone have a link to the board that we were working on?
No, sir, we don't have that.
I said we did do nitrogen compounds last time, right?
Yes, sir.
Yeah.
I'll just open, not really sure where the board went, but I'm going to try and. As the thing we had to do was, we did the whole disonium salt. No idea where the board went, but we did disonium azodize, etc. We did that stuff. And what we're going to do now is we're going to do a thing called proteins. Okay? So this is something that we are going to do. And we're going to do polymers. Okay? So these two things. So let's start with proteins. And with proteins, what you need is when you do proteins, the first thing that you have to study is the amino acids. What are amino acids? Okay, that's the first thing. Just a second. So you're going to study amino acids. Now, the list of amino acids is given in the data booklet that you have. An amino acid has this structure. It's got a carbon atom, and that carbon atom is bonded to two groups. On one side, it's bonded to a carboxylic acid, and on one side, it's bonded to an amine. So there's an amine and there's a carboxylic acid. So it's bonded to two things. And on the top, there's an H. So all amino acids have exactly the same structure, except for one difference. And that one difference is that the bottom is different. At the bottom, there is an R group. And that R group could be anything. It could be any R group. So you've got, the only difference between all amino acids is that the R group is different. And you've got a list of R groups that are given in the data booklet. Like if you open the data booklet, so somewhere in the data booklet, here's the names of all the amino acids, some of the amino acids, not all of them. They're actually, the human being is made out of around 19 to 21 amino acids. Those are our basic building block. So you've got alanine. In alanine, the R group is CH3. Alanine is known as by its 3-letter abbreviation, which is ALA, or its 1-letter symbol, which is A. So each one of them have a 1-letter symbol or a 3-letter abbreviation. The 3-letter abbreviation is kind of simpler because it's just the first 3 letters of the name. So let's draw alanine. The R group or the R chain is CH3. So that's going to be, if I draw that, what I'll just do is I'll copy this and I'll simply change what this R stands for. So I'm going to change it to CH3. That's it. So if I change it to CH3, that's my alanine amino acid. That's it. Similarly, I can change that to an aspartic acid molecule. That's another amino acid. So if I just copy paste this, an aspartic acid molecule is going to be this one. Where's aspartic acid? This one, the second one. It's CH2, COOH. So that's what aspartic acid is. So I'm going to drop off this R and I'm going to change it to CH2COONH. So it's basically a carboxylic acid in the side chain. And you can also have lots of other things. For example, lysine. This one is aspartic acid. Let me write that down. That's just the name of the amino acid. Then you have lysine. In lysine, what you'll have is that the R group is going to be different. In lysine, the R group is what? Open this. Look at lysine. This one is lysine. The R group is 4 CH2s with an NH2s. So that's your R group. So it's basically 4 CH2s. So it's CH2, another CH2, another CH2, and then there is an NH2 at the end. So that is what lysine stands for, or LYS. Is this clear?
Yes, sir.
Yes, sir.
Yes, sir.
So those are amino acids, right? Now, amino acids, they tend to link up. Like your DNA has all this information. Your DNA is this big, large strand. And we have to do DNA as well, but I'll just give you a basic idea of what a DNA is. Your DNA is this large coiled up ladder-like strand. And it's got all these bases that are emitting from it. Every cell has DNA in it, in its nucleus. You get the chromosomes and you've got DNA. Now, I'll just give you a basic idea. What the DNA, the information that is stored in the DNA is that every molecule in your body, like proteins, and I mean, they're basically proteins. I mean, your cells are proteins, your red blood cells are proteins. Everything is kind of made up of proteins, and proteins are made up of amino acids. So what the DNA basically stores, the information that is stored is that it stores information about which amino acid comes in what position, and how do they link up. That is the information that's stored in your DNA. So for example, your body wants to produce red blood cells. Now the red blood cells are going to be made out of proteins. So what the DNA would do is, the DNA is the one where the order of the amino acids is given. Like which amino acid comes in what position, that is determined by the DNA. So that's the basic idea. That's why the DNA is so important, because the DNA contains every information about which protein is going to be made in what way. So you've got very few building blocks, just 19 amino acids. Every cell is just a rearrangement of all these 19 amino acids. It's a combination of those 19 amino acids. So all that information comes from the DNA. All that information is stored in the DNA. Whenever your cell replicates, I mean, the DNA is the thing. I mean, the whole human body is actually working on the DNA. Like everything that has to be made, it has to refer to the DNA, like which amino acid would come in what position. But anyway, so let's say that the DNA states that you've got an amino acid, and there is an alanine molecule which is connected to a valine molecule, which is connected to, let's say, a lysine, which is connected to a cysteine, and you've got a C at the end. First thing, what is this N that I wrote, and what is this C at the end? Now that's just convention. Why do you need a convention? Because if you look at an amino acid, there are two ways to actually draw this amino acid. You could keep the N terminal. This is known as the N terminal. This is known as NH2. You can keep it to the left, and the C terminal is the carboxylic acid terminal. Every amino acid is going to have these two things. There's going to be an N terminal, which is known as the NH2 group, and there's going to be a C terminal, which is known as the COH group. Now, there are two ways of putting this. What I can do is I can sort of rotate it. And what I can have is that the carboxylic acid could be on the left and the NH2 could be on the right. But the convention is, so following convention, the convention is that you always keep the N on the left side and the carboxylic acid on the right side. Is this point clear?
Yes, sir.
So basically this N terminal and the C terminal basically indicates that you have to keep the NH2 on the left side and the COH on the right side. If it's the other way around, you'll kind of get it, because like if this is drawn the other way around, so that would kind of indicate to keep the NH2 on the right side and the COH on the left side. So we're just following convention. We'll keep the NH2 on the left side and the COH on the right. But anyways, there are 4 amino acids that are connected together. So let's pick a general amino acid. But let me draw a general amino acid first. I've got a C. It's going to have, there's going to be a carboxylic acid. There's going to be one bond on the top, one at the bottom. So there's going to be H over here. There's going to be C double bond O and OH over here. And there's going to be NH2 on this side. The only difference between the 4 is that the R group is going to be different in each case. So let's put an R over there right now. I'm going to fill in the R later on. So there are 4 amino acids. So that's the first one. Here's my second one. Here's my third one, and here's my fourth one. So I've got these 4 amino acids lined up. I'm going to change the R group now. First one is alanine. That we already know that is CH3. So alanine is CH3. What about the next one? The next one is valine. Valine is, open the data booklet. and you'll have valine over here. Valine is this CH with two CH3 branches, so that's valine. So I'm going to just change that. It is CH with two CH3 branches. And after that, I've got lysine, which we already did. That was 4 CH2s followed by, so I'm just going to put CH2 in a bracket and put 4, and I'm going to add NH2 at the end. And then I had I think I had cysteine. So let's find out what cysteine is. R group. Open the data booklet. Where do you see cysteine? The third one. That's CH2 with SH. So you got a CH2. You got a CH2 with S with NH. So those are 4 amino acids, which are mentioned over here. Alanine, valine, lysine, and cysteine. Okay, is this clear? 4 amino acids all lined up.
Yes, sir.
And what's going to happen now is they're going to start linking up. It's going to be a condensation reaction. Let's bring them closer. So all 4 amino acids, you did this in O levels as well, they're going to link up. And how are they going to link up? They're going to link up in this way that the OH and the H are going to form water. It's going to be a condensation reaction. OH and H are going to form water. OH and H are going to form water. So it's going to be a condensation reaction. Copy everything. Remove the H and the OH. So let's say I've copied everything. I'm just going to start removing the H and the OH from the middle. Right? They're gone. They've ended up forming H2O. So all of them, they've ended up forming H2O. And it's a condensation reaction. Okay, so this is the point that we have reached at the moment, and let's bring them a bit closer now. And they'll now start linking up because they are now short of bonds. All of them are going to start, they're going to start linking up with each other. So let's say they start getting connected to each other, and these are known as amide links, which would be called, in biology, they're called peptide links or peptide bonds. So this one over here bonds with this one. So this over here is referred to as an amide link. So this over here is a amide, or also referred to as peptide link. So that's the first thing. And this entire thing is known as a dipeptide. This entire thing would be referred to as, like, the whole thing. That's a dipeptide, not a polypeptide, sorry. A dipeptide would have 2 amino acids. This one would be referred to as a polypeptide chain, or a polyamide chain as well. You won't call it a polyamide because in biology you call it a polypeptide. I say, anyways, is this clear? This thing, they'll ask you to draw this. Is this clear?
Yes, sir. It's clear.
So that's, yes.
So do you have to say that there's 3 water molecules produced?
In this case, you're getting 3 water molecules getting produced. I said, but remember, a polypeptide chain usually is pretty long. It's not like every small protein. It's got lots of amino acids, not just 3. And it's going to be, there's going to be a repetition of amino acids. You could have a protein that will just have alanine. It would be alanine followed by alanine followed by alanine. Then there might be a lysine, then alanine. So it's basically, you can have infinite number of combinations of different, like 19 amino acids could be arranged in infinite number of ways. and the repetition is allowed as well. So you could have 1,000 amino acid chain, and there could be all sorts of combinations that you can have. So that's the first thing. Now, this thing is known as a primary structure. The one that we have drawn. Remember this, the human body stores all this information, like which amino acid comes in what position. It stores it in the DNA. Now, this first structure is referred to as your primary structure. Those taking bio might be familiar with this. So that's known as a primary structure. A primary structure is what information does a primary structure gives? What it gives is it gives you the basic idea of the arrangement of amino acids. So it provides you the order of the amino acids in the chain. In the protein chain. Is this clear?
Yes, sir.
So that's the basic structure of what a protein looks like. Okay, Baki Lubera, is this clear, Shafak? Zoha, is this clear?
Yes, sir.
Yes, sir.
So now there's another thing, and that is a secondary structure. So remember, it's like a thread. I mean, think of a protein as this long, tiny chain. I mean, in reality, the protein is going to look like this. It's like, I mean, like your fibers in your clothes, they're like threads. I mean, they're also polymers, macromolecules. So think of the protein as this really, really long thread-like structure, a really long chain of molecules all interlinked with each other. So that's what a protein looks like. Now, it's not necessary that the chain is going to remain. I mean, the chain is not going to be a straight chain. Like we've drawn a straight chain, right? You know that if you have a thread, the thread never actually remains straight. It coils up. For example, if you take this long thread, a sewing thread, for example, it's not going to be just like 100% straight. What will happen is it's going to get coiled up. That's known as a secondary structure, that the protein that's known as the secondary structure, that the primary structure starts to coil up. So the primary structure it starts to coil up. And there are two ways. I mean, the thread that you've formed, the long chain of thousands of amino acids that you've formed, it coils up in two ways. One of them is like a spring-like structure, which is known as the alpha helix. That's known as an alpha helix. Or it forms a sheet-like structure that the thread coils up in this manner. And that's known as a beta pleated sheet. That's a beta with the Greek P alpha with the Greek alpha. So you've got an alpha helix, you've got a beta pleated sheet. So if you can just Google this, maybe you'll find some. So alpha helix protein. So if I can show you an image, so you'd realize that this one is a good image. So you can see this is the protein structure. Maybe this is not that. Just a second. So that's kind of a good structure. So you just copy that. Notice I'm going to ask you about to draw the whole thing. That's slightly more complicated. So I'll just put this over here just to show you what an alpha helix looks like. So the chain that you thought was a straight chain, it's not actually straight. It's got these amide links. So you can see, I mean, I've written R groups. So they've actually just put R groups over there. So it's the same chain. I mean, you can clearly see that there's C double bond O, NH, then there's a C double bond O, and then there's another NH. So C double bond O, NH. So these are amide links that are just continuing. And the carbon in the middle are the ones that are with R groups. Okay, so the carbon over here is within R group. The carbon over here is also within R group. So every time you have C double bond O NH, then the carbon is within R group. Then C double bond O NH, then the carbon is within R group. So is this clear? This structure.
Yes, sir.
So, and the reason why it coils up is that there is hydrogen bonding going on. Like if I draw hydrogen bonds on this, remember the chain had what? It had C double bond O and NH. I mean, the protein chain has a lot of amide links. The bottom one will also have something similar. It's going to have NH followed by C double bond O. So what's going to happen is that the oxygen, which is slight negative and it has lone pairs, it's going to be attracted to the positive carbon atom. And there's going to be an attractive force between them. So the reason the chain starts coiling up is because of hydrogen bonding. This amide attracts this other amide, like there's an amide at the front as well. So the amide over here will attract this amide, so the chain would start coiling up. This thing will start attracting this thing, so it's going to start folding up. The same goes for beta pleated sheets. Like if I can just so this is what a beta pleated sheet looks like. That you'll have a bunch of I mean, this looks fine. So we'll just copy this one. So that's how a beta pleated sheet would look like. In this, the chains are kind of straighter. You got a chain, you got another chain. Okay, so it's probably the same chain that's kind of getting coiled up like this. And the chains are pretty straight, not coiled up in that fashion. They're lying flat. The NH over here is forming hydrogen bonds with the C double bond O at the top. The H over here is attracted to the lone pairs of oxygen over here. The oxygen over here, lone pairs attracted to the H. Oxygen over here, lone pairs attracted to the H. So there's hydrogen bonding going on between the chains. So this one is a lot more, it's like a sheet. I mean, that's why you call it a sheet. And it's less flexible. So the thing is, this one is flexible. Alpha helix tends to be more flexible, more spring-like. More stretchable. Beta pleated sheets are less flexible. So why are they less flexible? Because remember, when it's coiling up, you don't have a lot of hydrogen bonds going. Like the top over here and the bottom over here, they're not forming a lot of hydrogen bonds. So you can always kind of, you don't have to, like if you want to pull this upwards, not a lot of hydrogen bonds are between them. This one over here and the bottom over here, they're not forming a lot of hydrogen bonds. But in beta pleated sheets, one chain has forming hydrogen bonds with a lot of hydrogen bonds with the other chain, because the chains are lying side to side. So which is why it's not going to be very flexible. Like if you want to pull this chain upwards, it's going to be, you have to break a lot of hydrogen bonds. Is this clear as well?
Yes, sir.
Yeah.
So you've got all sorts of proteins like your hair, they're not flexible. So that means you've got a lot of beta pleated sheets in them. Your nails are not flexible. So that means you've got a lot of beta pleated sheets in them. Your skin is very flexible. It's very spring. It's very spring-like. It's got this stretchiness in it. You can stretch it. So that kind of refers to it having a lot of alpha helixes in it. Now, a protein molecule is going to have both. It's not going to be one of them. It's going to be both of them. A protein molecule would be containing both of them. So this is what protein with alpha helix and beta pleated. So if I can just find an image, or I'll have to draw one. So this is, I mean, that's one they've drawn. Just a second. So I'll just draw one over here myself. So an actual protein kind of looks like both. It's going to have both things. So the chain would be coiled up in this way. It will have beta pleated sheets. And then all of a sudden it might have a spring-like structure. And then it might have more beta pleated sheets. Then more beta pleated sheets. It's going to be a combination of alpha helixes and beta pleated sheets. So this is how the chain is going to get coiled up. It's going to have a bunch of both. TK, is this clear as well?
Yeah.
Now that's your secondary structure. Then you've got a tertiary structure. Tertiary structure is that, I mean, the tertiary structure is kind of the same. It's basically a folding. So we'll just quickly wrap up the tertiary structure. It's the folding of the secondary structure. The secondary structure will get folded up. So it's the folding or the further overlapping of the secondary structure. Due to R groups. Because remember, there are lots of, like, it's not just a single thread. It's got lots of these R groups. It's got a methyl getting out. I mean, the chain has a methyl coming out of it. It's got an amine coming out of it. It's got CH2SH coming out of it. So it's got lots of things that are coming out of it. Like you can see over here, there are lots of R groups. He even drew R groups over here that he didn't show them what they were, but you know, there are lots of options for R groups. So the tertiary structure is that the secondary structure that you just made would coil up or fold up on its own. Why? Because the R groups might be interacting. So it might, I mean, the alpha helix might be like this. It might fold up. Why? Because an R group over here, like if there's an amine, and on the other side there's an R group which is aspartic acid, and that's a carboxylic acid, what will happen? The two are going to react together. They're going to form an ionic bond. Salt would be produced, NH3+1 and CO-1. So they would be really strongly attracted to each other. Or there might be van der Waals forces, like there might be a CH3, and on the other side there might be valine, which is CH3 and CH3, and they'll all be attracted to each other. So all sorts of attractive forces are possible. You can have ionic bonds, you can have van der Waals forces, and there are all sorts of R groups. The R groups can get together, like there might be a carboxylic acid and alcohol coming from the other side. They might end up forming esters. So there could be all sorts of R groups. You can see that there's carboxylic acid. The R group contains In serine, it's got an alcohol as well. So they might form esters as well. They might form hydrogen bonds. Like if there are two OH groups, then they can get together to form hydrogen bonds with each other. So the R groups are going to be interacting with each other. So if you can see the tertiary structure of a protein, I mean, this one is a good image. Neither, not that one. Probably this one. So you can kind of see. So let's copy this one. So you can kind of see what's going on. That you had these beta pleated sheets and alpha helixes. And what's happening is that the R groups are kind of bonding with each other. A group over here might bond with a group over here. For example, there's hydrogen bonding OH, CO-1. So all sorts of ionic bonds, etc. they might be formed. So there's going to be further folding up of the structure. And they would get interlocked with each other. I mean, all the chain, the thread-like structure, it's going to interlock with each other. Is this point clear?
Yeah.
Yes, sir.
So, so there are three types of structure you've got, and they're going to ask you about each one of them. Primary structure, they might ask you to draw the primary structure. That's just the first one, the arrangement of the. So that's the first one. They're going to ask you draw this. That's the first question. Then they're going to ask you, I mean, starts coiling up because of hydrogen bonds. We need to write that. This is specifically for hydrogen bonds. So this secondary structure forms because all the amides are attracted to all the other amides. So the chain kind of starts coiling up, and it's because of the hydrogen bonding that's happening between the chain itself. And then you've got a tertiary structure that all the R groups that were not doing anything, they're going to start bonding with each other. So the chain would further start coiling up. So the end result is that the long thread that you thought of was a straight chain is not a straight chain. It's actually really, really coiled up, folded. It's folding in on itself. And one part of the chain is bonded to the other part of the chain. So it's all jumbled up. It's a globular protein, and they're all bonded to each other in some way. Okay, so that's your tertiary structure. They're going to ask you to draw this. They might ask you to show one type of attractive force between the R groups. So you can kind of show this, or you can show two alcohols forming hydrogen bonds between them, or an alcohol with a carboxylic acid forming an ester link between them. So let's continue tomorrow then, okay?
Okay, sir. Love this.
Chale. Okay.
Office.
Love is.